Here in part 3, we're going to talk about microbial metabolic diversity. Central metabolism involves both catabolic pathways, in which molecules are broken down, and anabolic pathways, in which molecules are made. Diversity in the energy generation of catabolic pathways and anabolic pathways is a key to the high diversity of metabolic pathways that we find in prokaryotes. Let's start off with the beginning. All organisms can be classified into either those that are phototrophic, that is that they get energy from the sun, or chemotrophic, that is that they get energy from chemical compounds. All organisms need that energy that's used to generate ATP to conduct different life-associated reactions. All organisms need carbon compounds that serve as the building blocks for cellular components. So if we begin to look further at metabolic diversity, we can take the autotrophs, those that make food from CO2, and distinguish them from chemotrophs, those that can't make energy on their own. We have photoautotrophs that take carbon from inorganic sources such as carbon dioxide. Cyanobacteria and vascular plants are an example. And then at the other end we have chemotrophs. They get energy from compounds. We have heterotrophs such as most bacteria and animal systems such as you and I. We can diversify this a little bit more by separating phototrophs into the autotrophs that use carbon dioxide and sunlight to produce compounds, versus those that use a different carbon source plus sunlight to produce their organic matter. On the other side, we have chemotrophs that can be defined as heterotrophs, as we just discussed, or chemoautotrophs, those that take carbon from an inorganic source such as CO2 and reduce it and use that as their energy source. So at this point, we'd have a clicker question to touch the photosynthetic organism that can't use CO2 as its source, and those would be, of course, the photoheterotrophs. We find some very interesting metabolism when we begin to look at chemoautotrophs. Here's an example from Bill Brazelton looking at tube worms that live in hydrothermal events. They've replaced their digestive system with a microbial bioreactor. That is, that they let microbes conduct the reactions that you might expect to find in a digestive system. So carbon dioxide and oxygen come into the system, and so do sulfide ions. Here within the internal part of the tube worm, the sulfide and oxygen are combined to produce sulfate. The energy which is released from that reaction is then used to generate the biosynthetic reactions to make organic molecules. So this is an example of a chemoautotroph. If we begin to look at microbes in very harsh environments, we see some very, very extreme situations, such as these mine tailings that you'll find throughout much of the West. They have very low pHs. They can actually be quite warm if they're associated with a thermal vent, and high in iron, zinc, copper, and arsenic. If we begin to look at the DNA sequence or the RNA sequences that'll come out of here, we find that there are both bacteria and archaea present. We find tremendous diversity in the archaea and bacteria present, and you only have to look at the human to see this diversity. Here we're representing that diversity with different trees of life so that you can see the complexity of the archaea and bacteria that are present in your mouth. versus those in your airways, those on the surface of your body, those within your intestines. And notice that there's a difference between what's in your mouth and in your intestines. And if we were to look at vaginal regions, we'd find yet even a different set of microbes supported within our bodies. Now, just for perspective, it turns out that the number of genes associated with humans and the number of genes associated with some of our microbe communities, and here I will use the gut microbe community for comparison, is quite different. There are only about 20 to 25,000 genes in the human genome, but in our gut microbiome, we have over 3 million gene combinations. If you can take the time, I suggest that you read a book published by a wonderful writer, Ed Young, titled I Contain Multitudes. This is a description of what life is like in our gut microbiome. It's a fascinating book to read. Here's some surprising facts about our gut microbiome. We have an almost equal ratio of eukaryotic and prokaryotic cells. But because there is much greater prokaryotic diversity than eukaryotic diversity in our gut microbiome, there are more prokaryotic genes. These prokaryotes include Archaea, that as a sink for hydrogen and produce methane, and bacteria that take up sulfate and produce hydrogen sulfide. That's why when humans flatulate, we can actually release methane gas and have a smell such as you would find in rotten eggs. We call the community that is complex and within our gut biome a holobiont. It's a mutualism, and it involves a physical, chemical, and metabolic series of adaptations on the part of the host and on the microbes. Now, at this point here, let's introduce a concept that might be familiar to you. It's called ecology. And ecology is the study of interactions among organisms and their environment, or among organisms that are interacting. So we can have both a biology interaction or a biology physical environment interaction. Now, you could use the phrase that one microbe said together, let's hang out. And it turns out if we begin to look at the diversity in archaea and bacteria that are in our guts, we often find that the input from one organism is actually the metabolic waste of another, so that we can categorize energetic life using a framework to understand how this diversity fits together. And one easy way to do this is the electron tower. Now, it's been said if there's a living to be made, microbes will find a way. We find that if we consider different donor electrons for a reaction and different acceptor reactions, we'll have different waste products. So there are organisms, methanogens, that take diatomic hydrogen, link it with carbon dioxide, and produce methane, and along the way extract energy. We have organisms that take diatomic hydrogen and combine it with sulfur, and along the way produce hydrogen sulfide. So we have a whole series of chemical reactions that can take place, so that the waste product of one microorganism is actually the source for another set of microbes.